time alone is a risk factor.
Over time, as we age, we accumulate mutations that can cause cancer. Our body lose the ability to repair those mutations.
What were you thinking when you first took your research on chain?
I certainly was aware of Bitcoin early on, but this whole idea of leveraging it for science, developing drugs was not on my radar at all.
It is your research and your project. There should be an incentive to make it big.
If it works, then that whole $3 trillion thing could actually be at play with what we're doing.
Hello, everyone. Welcome back to the DeSci Podcast. Today, not with your usual host, Ella, but with me, Alana, and our guest, Michael Torres. Welcome, Michael. Pleasure to have you.
Thank you. Pleasure to be here.
So Michael is founder of Atan Bio, a biotechnology company developing a codon suppressor therapy for aging biology. And Michael has made the choice to take his research on chain. And today we will talk a lot about your backstory and tokenizing your project and everything that comes with it. I think for those of you who follow this podcast, you've seen we spoke a couple of weeks ago and we touched upon the current stages your project is in and where it is going. But maybe for everyone who might have missed this, you can just briefly introduce yourself and your research and what stage you're currently in.
Sure. So as Alana mentioned, Michael Torres, one of the co-founders and CSO of Atan Bio. I'm also the CEO of an VC-backed company called Crossbridge Bio, which is firmly in the traditional biotech space. We've got traditional investors. We're, going to be in the clinic next year in the oncology space. And then prior to that, I also co-founded a company called Recode Therapeutics, which is a genetic medicines company. So that's RNA medicines, nanoparticles. That's raised, $400 million since inception. And I was one of the original scientific co-founders of that company. And then has 2 assets in the clinic right now for rare respiratory diseases. And then prior to that, I completed a postdoc and PhD in cancer biology at UT Southwestern in Dallas, Texas. And before that, I had actually gotten a scholarship from the NIH and spent time doing research at the National Cancer Institute, focusing on DNA damage, DNA repair. So all of this sort of circles into, what we're doing at Atan Bio. So from my personal background, you can see I've sort of been in
many different roles
of drug development, different projects. So I'll definitely touch on Atan a bit further. Maybe just on the funding history, and then we can dive in deeper to any aspect that you want. Right. This project came through VitaDAO, where I was an entrepreneur, sort of company builder. Working with VitaDAO in around 2022. And helping kind of think through projects that they would fund. And then I had my own project that I had been thinking about for a long while that I was able to put into VitaDAO. So we raised about $100K to do some early experiments. And then,
and that was the project VitaRNA we're currently talking about, right?
That's the VitaRNA project. So Atan Bio is the company that we created the project that eventually went into VitaDAO as now a tokenized project, VitaRNA, which it's a longevity-based asset, longevity-focused asset, looking to significantly expand, healthspan, lifespan, and what have you. So again, happy to touch on all of those, but maybe that's sort of the
more into detail into that later, but maybe more about the main goal of this research project. Maybe you can explain what are these nonsense mutations, what is a codon suppressor, and why is it actually important?
Yeah, so I think maybe to just kind of level set with folks, I think it's well appreciated that over time, as we age, we accumulate, damage, we accumulate mutations that in some cases can cause cancer, which I think is sort of the biggest, easiest thing to recognize, that over time, whether it's pollution, genetics, UV exposure for, melanoma as an example, all of these things typically cause mutations. And naturally, over time, our body also, can sort of lose the ability to repair those mutations that occur. And so then you can have, again, this aging effect that can be driven by, these mutations that can occur naturally, because our body also, isn't 100% accurate in repairing mutations. And again, your cells are dividing all the time. DNA is being replicated. This process is happening all the time. So in the case of what
So
Artan
just
and
time
development,
alone is a risk factor.
yeah, exactly. So I mean, it's we all get older today. We
Yeah.
all age, and eventually we all die. Like that's natural course of events. And so the basic premise of Artan, and again we can drill down in specifics, is that we aim to slow down that process of acquiring mutations that happens. And so if we're able to do that, and particularly in important proteins, and these are the the things in our body, proteins are that function, they do all the work in our body, that if we can basically keep all of that up in high levels of repair proteins, keep our repair really high, keep the ability to suppress cancer, because we have these natural proteins called tumor suppressors that are that we all have, that help do what it says, suppress cancer. So if we can keep all of those up for longer, that potentially could significantly increase our lifespan, because again, we're not, those things don't go down over time. And so one way that happens in a really interesting manner is that, there's a particular amino acid, and an amino acid is part of the building blocks that make up proteins. There's 20 amino acids in humans. One particular amino acid is very sensitive to mutation. And in that case, we are now looking to essentially restore that particular amino acid, keep it present high, and so that even if it were to get mutated, we're sort of putting back that amino acid in very important proteins. And what's interesting, evolutionary, is that when you look at this particular amino acid, arginine, and then arginine has different codons. Codons are 3 nucleotides, and nucleotides are DNA that make up what arginine can be. So again, there's like, I believe 6 codons that can be used to make the amino acid arginine. Okay. And one particular codon, CGA, again, this C, which is called cytosine. I know we're doing a lot of science here now, but cytosine is sensitive to being mutated. And so when people think especially about aging, and you've probably heard about, methylation clocks, aging clocks, it's typically driven by cytosine methylation. And so that then typically reduces transcription levels, which means that these proteins and the mRNA and all of the RNA molecules are silenced. If they're silenced, then they're not around, and then they don't do the work that they're supposed to do. Okay, so the cytosine is a very important nucleotide. It can be methylated, it could be deaminated. All of these things are basically mutations to cytosine. And so if you change that cytosine, or it gets mutated, it can be either thymine or uracil, which are other nucleotides that then, instead of a CGA, it can make a UGA, and a UGA is a stop codon. And stop codons, they say, they tell the machinery to stop making the protein, making the RNA. And so you can imagine that if you start putting stops all over wherever these arginines are, that could be a bad thing. And so if you look where arginine CGA codons, again, there's 20 amino acids, and each amino acid has multiple codons that can make that amino acid. CGA codons are actually way underrepresented. So what that means is that through evolution, we have tried to get rid of these CGA codons because it's deleterious, right? But we can't get rid of all of the CGA codons because, again, just evolutionary, they're there, they're needed.
I mean, if they're not bothering, they're not being eliminated also, right?
Yeah.
That's
Right. But that also makes, again, there's certain proteins like DNA repair, tumor suppressors that still have these arginines present with the CGA codon. And sometimes they have multiple CGAs, which again means that hopefully just if one's mutated, they have others, and it could maybe still make a protein enough that could still be functional. But the point is, the body, or, we've already been trying to suppress these. They're still there. And so now it makes it a very sensitive situation where one on those could be really consequential, because now, what's left are the things that really need these arginines. You can't get rid of it, basically. It's another way to think about it. So all that is a long way to say that VitaRNA, Atan Bio, we've developed an approach to suppress the functional outcome of those mutations. And so in this case, anywhere where those CGAs are left, and again, they tend to be in very important processes related to aging, DNA damage repair, tumor suppressors, proteins involved in neurocognition, so that when they go down, they're associated with Alzheimer's and other things like that, Parkinson's, that we can put this codon suppressor in and keep all of those proteins up for longer. And again, hopefully really, really significantly address lifespan and health span, because then, your cancer incidence could go down, the potential to get Alzheimer's or Parkinson's could go down. Again, as I mentioned, your DNA damage response could stay up. So then, you slow down the rate in which you accumulate mutations, which are associated with the gene. And that's the big idea that we're trying to work on. And we're leveraging DeSci and crypto and all the fun stuff to do that, because, traditional pharma and biotech, has a challenge thinking about these types of aging concepts. Now it's changing a little bit. There are now typically driven by tech investors, tech guys who want to live forever. They're funding these longevity companies, but traditionally that's been a very hard sort of area or therapeutic area to focus on by traditional VC and pharma. So that's where DeSci has really stepped up, and it's exciting to see the progress that's being made, leveraging, the alignment of this community.
Yeah, definitely. And I think your project is a great example because your timelines, they speak for themselves. You took 3 months to go from proposal to initial raise, and then 6 more months for the second raise, where you accumulated more than 300K. And now, as you said as well, there are some updates also for your research, which we briefly touched upon a couple of weeks back, but maybe you can just explain again where you're currently at, and maybe if there is some news as well.
Sure. So yeah, I think that's again what's exciting about DeSci, particularly in contrast to funding, other funding mechanisms like VCs or grants and what have you. That the process can be very drawn out, and here it was relatively quickly. I think it still could be a little bit more efficient as somebody who is looking to continue to build this project out, but nonetheless, it's certainly much more, the speed is just much quicker than these traditional funding sources. And what's really nice about this path, and so to touch on the progress that we made, as you mentioned, right, we got an initial funding to take these concepts, which were just like vector designs. Again, this is a gene therapy. We think that's one of the primary sort of ways that this could be the most effective, is if we could have, it expressed in, as many cells as we can get in the body for as long as we can. And so, but these were just designs on paper. And so, just the fact that we could pitch that idea, get some money to make those vectors real, and then test them. And of course, I knew the models, the experiments to do. Like, we were able to do that very quickly. And so what's also, I think, really nice, and in contrast to traditional like biotech builds where you're very quiet, you're in stealth mode. I see so many companies that are in stealth mode.
Yeah.
Right. And I'm like, okay, I guess that's cool. It's different. In DeSci,
Yeah.
you want to show what you're doing. You want to show the community, like, being open and transparent is such a cool feature and an enabling feature in DeSci that...
And it's being rewarded instead of being punished.
Exactly. It's rewarded. Token holders can see what you're doing. We use Telegram and Twitter a lot, so that we can talk through what we're doing with the community, try to be as transparent as possible. Of course, intellectual property is important, so we keep those things as walled off as we need to. But all that to say, we're very open, and so again, that's what enabled the the sort of tokenization to 300K that we raised, to then further take those into these vectors into animal studies, and typically, we did, typically you do like mice, rats, then maybe non-human primates. That's like the traditional drug development path, where you can get some early safety and efficacy in rodents, and then eventually you move into non-human primates. And then most regulatory bodies, i.e. the groups that will allow you to do a clinical trial, legally, will want this data package that you put in front of them. They review it and they say, okay, this looks safe enough to go into humans. And so with that 300K, we made our product, Atan 102, in a way that is scalable to support human studies in the future. But of course, you have to scale up over time. So the next, monkeys are bigger than rats, and mice, so you need to scale up the amount of material. So that's one of the most important parts that you need to do, and have all the associated analytical assays. This is, again, gets very technical in drug development path, but the point is, we started working on a process that could manufacturing process that could support these larger studies. And then we also looked at some preliminary safety to make sure that at the dosing that we think we ultimately want to get to, that there's no red flags, anything that's really stood out that we need to adjust. And so that was what that first 300K helped. And then now we're continuing to raise even more to support a non-human primate study. So in monkeys, where what's called like a non-GLP tox study, in GLP means good laboratory practices. There's guidelines, again, that regulatory bodies want to see. And so what you typically do is you'll test in a non-GLP setting, you get your dosing, get everything sort of worked out in a smaller study before you do a larger GLP tox study. And that study then typically, along with your manufacturing, is what supports a human clinical trial. And so we're now working towards generating that material for a non-GLP study and non-human primates. Hopefully we'll be able to do that by the end of the year, and then early next year do a GLP tox study, and then hopefully a clinical trial. Now, what's interesting about DeSci and sort of this longevity space that we're in is there are, you know, regions in the world, geographies that are embracing longevity. So, you know, UAE, there's Montenegro, there's other countries and jurisdictions, Kazakhstan actually, as well, where they want to work with companies like us to help design those GLP tox studies in a way that then right after that, if it looks good, you can start a clinical trial quicker. So, you know, you see that the traditional sort of path to a first in human, as we like to call it, can be much more streamlined when you sort of line up the interests in a really efficient way. So again, leveraging geographies that have an interest in longevity, and then, you know, making sure that you do the studies that they want to see, that will, you know, again, assess the safety and efficacy, and then hopefully do things a lot quicker than traditional drug development timelines, and in an open way that hopefully the community is excited about, pharma's excited about as well, because they can see like, hey, there is a different way of doing
Yeah.
things.
Yeah, and it
Uh,
doesn't mean sacrificing any securities, any of these things that are happening in traditional research as well. It's
yes.
just like you said, you align founders' needs and the research needs more with what needs to be done and how to get there in a fast and an efficient way.
Yeah.
But all the security measurements and everything is still in place. So
Yeah. Safety is key. And again, you know, as you heard from my background, I mean, I'm been in drug development, multiple programs, you know, again, funded by, you know, traditional VCs, gone to the FDA. So, you know, I know the importance of doing things right and not cutting corners. You know, that, again, you can be lean and efficient without cutting corners. And that's what we're aiming to do with this project. And so, you know, there is a high bar for me on what that data package needs to look like so that, you know, it'll be safe going into people, because that's, you know, like nobody wants a problem there. You know, I certainly do.
yeah,
So we'll do it
okay.
right.
I would like to circle back for a quick moment. Just now we touched upon a lot of different things, also tokenization and DeSci and you discovering molecule and also that you can tokenize your research. But all the benefits and everything you talked about now, was that something you were aware of before, or was that something you have discovered on the way? And maybe just explain your main motivation in the first place. What were you thinking when you first took your research on chain, and what were you hoping to get out of?
Yeah, so you know that's an interesting question, and actually a very interesting story, at least from my point of view, where, you know, DeSci, crypto. I mean, I certainly was aware of crypto, particularly, you know, Bitcoin and other things early on. Um, but this whole idea of leveraging it for, you know, science, developing drugs was not on my radar at all. And in fact, you know, my first company that I had co-founded called Recode, one of the largest investors in Recode is Pfizer Ventures, and
Mm.
Pfizer had also invested in VitaDAO, and so it was actually my friends at Pfizer Ventures who got me clued into VitaDAO and this whole space, particularly as I was transitioning from Recode and I was looking
So why
for
did they
the
think it could be particularly interesting for you?
because I have a like drug development background, science background. You know, I've talked to VCs, I've raised money from traditional VCs. So, you know, in that case, my friends at Pfizer were like, look, they need more like company builders, people who have the experience that you have, uh, um, to pair with, these, the VitaDAO, everybody, they know the crypto, the Web3, that crowd. And it's like, okay, but they need more experience in like drug development and biotech builds. So it would be really great if you could like help them and help level up the organization with my background. And, you know, at first it was just something to do. I was like, sure, I have the time. Let me explore this a little more. And then, again, having been a founder, which, my first company is still a private company. I mean, I have a lot of shares, but I've never gotten any sort of upside from those shares as a founder of the company, and then particularly in biotech, there's this big debate about founders and first-time CEOs and who do you back and who's going to get the big VC dollars and what have you. And so in most cases, and I don't disagree with this and view at all, the big VCs are not going to, typically, back, someone who hasn't done it before. Like they want to see you have a track record because it's hundreds of millions of dollars, just to, potentially get to the clinic. There's so much that needs to happen. They want somebody who knows what they're doing. Okay. And so for me, with my first company, Recode, I didn't have that track record. And so, I had some of the VCs tell me like, look, we know you know good science, but, we're not going to back you because you don't have a track record. You haven't made us any money. And so like, there's a wall for a lot of founders that just exists. Folks break through, and I feel like on the traditional VC side, I've broken through, but there's this alternative. I think that's really exciting for founders and scientists, which is DeSci, where then, you have more ownership, you have more control, you have more engagement with the community. And in the case of a token, and particularly if you have a plan, a project, and you execute, there's opportunity for, upside in a way that doesn't exist with traditional VC backed, where, with VCs, founders are quickly diluted because it takes all these hundreds of millions, right? And so they want more and more of the company for that. And that's just the way the model and the system works. There's no faults against that. But DeSci presents a really interesting alternative where now founders, scientists can retain more ownership of their projects and also participate and partake in more of the upside of your successes. And so it puts your sort of opportunity much closer aligned with the outcomes of your work, which again, I over at 8-9 years at Recode, and I don't really have anything material to show for it,
Yeah.
just experience and all this sort of
Yeah.
stuff.
Yeah.
I'm not knocking the path, but DeSci presents something different, and I think that's exciting. And if you do it in a way, again, where you're transparent with your token holders, you're rewarding them as well, it truly can be, win-win and much more aligned with a smaller group that the sky's the limit, right? And that's what's, I think, really cool. One of the cool aspects about DeSci. And so again, as I started to learn that by working with VitaDAO and seeing the opportunity, I was like, hey, I want to put a project in.
Yeah.
And then of course, obviously you've seen the successes and what we've been doing. So I feel like we're a good use case for this concept. And, I will continue to, try to make that be the case for as long as I can.
Yeah. That's the best outcome we could imagine, anyways. I think now you also talked about the upsides a little bit, but maybe you can go a little bit more into detail of the specific upsides you got from DeSci and tokenizing your project you would have not gotten if you would have gone the traditional route with this project in particular.
Mhm. So, I mean, the obvious one, and I think it should be sort of front and center because, it is you know, sort of economic part of tokenization. Okay.
Yeah.
So, again, when you think about a traditional company that you build where maybe you have 1%, even less, and that is not, that's not even liquid. Like, I have shares in Recode. I can't sell them right
Yeah.
now. It's not public. There's a long path to, potential, likely just, let's just be real, very little upside for founders. Okay. So then you contrast that to, let's say, in academia where you're writing grants, and grants are there to fund science. You get a little bit of your salary, but if you want to sort of monetize your research, and especially if it can be, and not all research should be monetized, and there's room for everybody in science, it's very hard, and you eventually have to go through a tech transfer office, then have somebody license that, or you start your own company, you have to go raise money, and you're back to the VC world, okay? Where with tokenization, and whether it's like a specific project or even a DAO, where I've seen folks who have it like, hair DAO or spine DAO or any of these DAOs that have a specific focus. Now they've been other people within a community, or to build a community that then will put money into a plan, and with either those, like, the leaders of the DAO, or, in our case, Atan and VitaRNA, like the PIs or the researchers for the projects, they sort of put their money and their trust in this particular an area. And so then, right, in exchange, they get tokens, in ownership of that. The scientists get, a token percentage. And so in my case, I got an allocation of tokens for VitaRNA. And so I am now motivated, incentivized to make the project as successful as possible, so that the token price goes up, the token holders are happy, I'm happy, everybody's happy. And that just doesn't exist in VC, in academia at all, right? So this
Yeah,
is,
to a point where I see you feel a little bit hesitant to talk about this upside, right? But it's a real thing because it is your research and your project, and
yeah,
there should be an incentive to yeah,
yeah,
make
and
it big and
it's
get
that's
it out.
right.
And
And you're right to sort of see my hesitancy to talk about it a bit, but again, I think it. I agree. It should definitely be talked about more. It's a real an incentive, and that doesn't mean. I mean, drug companies do research for profit. Okay, like you get grants so that you have a salary for yourself in your lab. Like we're all we're all selling something. In the case of DeSci and tokenization, to me at least, it's the most aligned model that I've come across for researchers, scientists, stakeholders to all participate much quicker in any upside in what you do. And I think that's really cool. That's a really
yeah,
cool sort of a thing that exists. And maybe to speak to the community part just a bit as
yes,
well, because that's obviously very important too. So, like, it is really nice to have stakeholders there to access. So, like in the case of Crossbridge, my company, my stakeholders are my investors. In my case, right, they've put up millions, so it's a different level than token today. But the only time that I really engage with them is during board meetings. And yes, you can have one-offs and talk with your folks, and I have a great board, so it's really good that they are very accessible. But contrast to DeSci, where again we're very active on Twitter, and I'm a Twitter lifer. I'm not an X guy, but it is what it is. And
Me too.
Telegram, you can thank you. Uh, you can engage with the community much easier. Like we had
Yeah.
put in a request, like I wanted somebody to do some like bioinformatics work. So we asked, hey, could somebody, work on this? Here's the need. A few folks stepped up and generated, Excel,
Yeah,
files
yeah.
with
There's
things
general
annotated.
excitement also.
Like that's yeah. So the
Yeah.
ability to tap the community, because if you're doing it right, you're very engaged with them. They're also up to speed on what you're doing. Creates an opportunity. And Research Hub, I would say, is like another example of how folks are trying to align incentives with experts or just people that are interested in a particular area. And I think again that doesn't exist when you're in stealth mode as
Yeah, yeah,
a
yeah. How can it?
right, right. And for better for worse, maybe in their case, well, we don't want to tip off our competition to what we're doing, et cetera, et cetera. And then if you're VC and you've got to make returns for investors, like you have to, that makes sense,
Yeah,
So
yeah.
contrast to DeSci where it's open, it's transparent. I think it remains to be seen, how successful the model is. Again, I'm hoping and we're working, diligently to make sure that, VitaRNA is and Atan is a good, again, use case for this. But I think, yes, there's, we're going to embrace being open, transparent, getting insights from our community and see if we can build something even better than what we were thinking about originally.
Yeah, and I mean, I think it's also that's the core idea of research in itself, right? Like to reiterate and see where do we need to make changes and these things. Just from the community perspective, again, I think there's no other place where you can be in direct contact with researchers, where you actually get direct updates of what is happening in the lab, what stages are planned, what are the next steps that are planned, and everyone who has an interest in this research can actually be involved.
I think it
And
yes, go ahead.
please go on.
Yeah, well, just to reiterate or maybe emphasize a few additional points in this regard that we haven't covered is again like I, right we live this being very active. I mean I'm very accessible whether on Twitter or on Telegram. We have a VitaRNA token holders chat or Telegram thread. I'll also give a plug to Molecule and their and I forget the exact name, so I hope Kevin doesn't get too mad. But the well on molecule.xyz, actually the like labs or the screener. But then when you go and click on the Molecule Labs, where now you've got the updates that are present there, they're accessible. Anybody can go and look at that. I think there's multiple ways to engage with the with scientists to the community to the research, and all of that again facilitates a dialogue, discourse, or it can, and we certainly want it to, so that people will pressure test our ideas, ask questions. And maybe they're not experts, but they want to learn something and know something, and then they feel more empowered about like what they're investing in and the
Yeah,
ultimate outcome.
exactly.
So I think again, it's nice that Molecule has built out this infrastructure, but what I would say is that the best case is that it is a two-way street, right? So that our ideas can get pressure tested, they can get, we can get feedback on, and throughout the course of this project, we've certainly got our fair share of feedback. I was actually revisiting, I think the first, I believe it was on Discourse, the conversation about funding VitaRNA at the
Yeah.
beginning, and there's a lot of comments. We had gotten a lot of comments, a lot of questions, a lot of insights from the community before they ultimately voted to fund this. And again, it was really interesting to revisit, because I can see like the progress that we've made,
Yeah,
and
yeah,
to say for the skeptics that were there early on, who are now sort of fully on board and engaged and are like,
yeah,
yes, you guys are
yeah,
totally
yeah,
it. It's just a neat kind of thing where I'd say in traditional science, the way of getting feedback, and now things are getting a little bit better, would be reviewers when you're submitting articles, and we all know, especially those of us in science, about reviewers and who are peers, and certain takes that they can have. It's very interesting. And then typically at conferences where you present, you'll get live feedback, and so I think again the strength of DeSci, and I hope to see more of this get built, is the ability to have rapid sort of turnaround, real time dialogue on projects. To again, in our case, I think last time I looked, maybe there's close to about 200 token holders. Maybe all of those talk to us online, but they have the option, the ability to. And so you've got to imagine that the more and more we start talking, the more this, gem can be polished, and we're going to have a better, outcome, or at least a better probability of success of a positive outcome. Drug development is hard,
yeah, yeah,
but it's really great to have that. So again, I'm very much always open to receiving feedback on how to further improve these things. And so, I'll put the charge to the community and listeners to, reach out and ask, ask questions.
yeah.
Hey, tell me what you think. Tell me how we can do better. And we can be as responsive as we can to these things, or we certainly will be.
Yeah, it's amazing to
So
see also that, yeah, you're so open to the whole concept of this communal sharing. What I wanted to ask you as well is, now you talked about voting real quick, but maybe for the people who don't know what it means to vote, what governance means in this concept of IP tokens, and what it means to be involved in a project through governance.
yeah, so in our case, again, and I think each project has, and researchers have their own sort of English flavor of how they implement, but the whole idea behind tokenization, and one of the core ideas behind that is to be able to leverage the community for decision making, which in reality is another way for engagement, right? And so and in this case, we will put up certain decisions, and let the community vote with their tokens on whether we do X or Y, or various things related to that. And so again, I think in one hand, I mean, just being candid about it, right? Like, you're not going to put every decision out there for token holders to make, because again, like, we're drug developers, experts. We know the thing to move the things forward, and trust us, when we share data, it's going to be good and, well received, and hopefully, there's upside, appreciation of what we're doing. But I do think, again, and we've done this a few times already, certain decisions that we do ask the community to vote on, and certainly when it comes to the tokenomic side of things, like, hey, we want to raise more money, we want to and various things like that. There are we do need to go to token holders for that, and there's a membership agreement, right, between the token holders and the stakeholders on the tokenomic side that again govern how we make decisions. And so I think there's the layer of certain decisions that impact token holders have to be voted on, and then there's another layer of engagement decisions, maybe I'll call it, and we try to certainly make sure that we do the ones that we need to, but then how can we empower the community to sort of feel more engaged in what the project itself. And so I think, to be honest, that's probably an area where we could do better and do more. I won't say do better, I'll say do more. We can do more. Okay.
Yeah.
And so I would certainly put it to the community and your listeners to provide that feedback. What are the types of decisions that you would like to be a part of, or types of topics or areas, and we're happy to engage in that manner. So definitely open for feedback on that, for sure.
Yeah. Have you made this or do you think in general the people also give you feedback on these things?
In
What type of decisions they would like to have more governance or be more involved?
I don't know. I'm asking now. We've never really sort of asked that. We've never really asked that. So
Yeah.
we've primarily stuck with the token governance things.
Yeah.
We had a couple decisions around we set up a bounty for some work that we did, and so we approved some tokens to give as
Yeah.
bounties, different things like that. But there could be other things, and I guess this is me asking our community, the VitaRNA community, what other types of decisions do you feel, or questions would you like to be queried about going forward? Again, I think in my mind, I also thought about, as we start accumulating more data, we could go present at different conferences. So maybe we can put it to the community to say, okay, which conferences do you think are would you like to see this at? Deal. Of course, we'll weigh in ourselves, because I do have I'd say a bias towards getting in front of more traditional academic and company researchers conferences, versus I would say, at least for VitaRNA, the longevity space, there's some conferences that to be candid, feel more like Lollapalooza, that I'd
A
say.
little bit.
Almost like a festival, more like a
I think
Friday
they all are a little.
than a place to have your ideas pressure tested, have your where investors, traditional, I don't say traditional, but where investors will be,
Mm.
what have you, because a lot of times, right, so when you get very narrow in a field, and then it's just you're it's an echo chamber, right? It's all people think the same. They're not, and you're not going to get that well, this is why this will never work, or have you thought about XYZ, or hey, I developed this drug and this was the dosing I tried, and we ran into this like tox problem. If it's all academics talking about their own research and how they're solving aging, you're not going to get that, right? So I want to be in spaces where we can show what we're doing, but then also get relevant feedback on how we can like move things forward, as we're thinking about a therapy and not a science project, and that's just part of like, I think, where, that's just how my view is, having been in multiple companies and have started multiple companies, and I'm a, drug developer, not a academic PI who writes books about stuff. I'm a doer. I want to do it right, which is why you need feedback from folks.
Okay, I think maybe just to finish this conversation, is there one thing your IPT holders can look forward to in the next coming, or maybe in the next year?
Yeah, I mean, look, I really think that we have an opportunity to, have this longevity asset in humans.
Yeah.
And again, the sort of like, how do you address aging is still an open question, right? And there's like folks like Loyal that have done an aging trial. It's in dogs, right? And I think that they're going to try to use that to think, how to get into people. So, if you check out either VitaRNA.xyz or molecule.xyz and look at VitaRNA, we have our non-confidential presentation in there, where I think in talking with regulators, actually, we've come up with a strategy that can align an aging trial in an indication where it's very clear that aging drives like the disease, that we can go and test that. And if we can do that and that shows a clear like clinical benefit, then I think we have potentially unlocked, aging as an intervention that we or as a therapy that can have interventions, because that's still a big issue with how people think about aging trials. I mean, if you just look, how do you do a trial on aging, blah blah blah, there's all of
Yeah.
these things. And so what most traditional companies in the aging space have done is they go after a rare disease or like a particular aspect of aging, like arthritis, as arthritis as an example, and say, okay, we're going to use our drug here, and then maybe if that works, there's this larger market that is potentially accessible. But that's not really aging, going after aging. It's going after a disease, related to aging, but I would say not still proving that your drug can affect aging as a whole in
Yeah.
terms of lifespan or health span. So the strategy that we've come up with Atan 102 and, the VitaRNA project, I think actually does align with an indication or disease that's big, is almost as big as aging itself, unfortunately. But if it works, then one, that's like not a rare disease, very large, huge market, and then we will have proved the aging thesis, and so then that whole whatever $3 trillion we're all aging sort of thing could actually be at play with what we're doing, if it all works, and that's what's like really exciting. And so hopefully over the next year you'll see us making strides to checking off those boxes along that way, and I think that's pretty cool.
Yeah, definitely. Super exciting. I think that is it for today also. Thank you so much for your time. If you want to check out Michael's project, you can go on molecule.xyz /VitaRNA or VitaRNA.xyz. And
Yep.
what is the Telegram and Discord channel you have?
There's on VitaRNA.xyz, there's a link to the Telegram on a channel, and then maybe you can put up my Twitter name account on, the page and or what
Yes,
happened, and then share it that way, because I'm always happy to engage with the community that way too.
definitely, definitely worth to follow. And thank you so much for taking the time, Michael. That's it for today, and see you next week, guys.
All right. Thank you very much.
Bye
Bye.
bye.